In this video, we're going to continue to look at the function of DNA. And previously, we looked at what happens between generations of cells when DNA is replicated. But the other part of that is we want to take a look at, well, what does the cell actually use the DNA for? And so we'll take a look at this little bit here in terms of what's going on within a cell when it's actually using the genes that are on those chromosomes there. And if we take a look at the first step in this process, it's what's known as transcription. And this is sort of a play on words because when you transcribe notes, you're basically making a copy of them. And that's what's happening when we're looking at our DNA is we're going to make a complementary RNA copy of one strand of our DNA through the process of transcription. And when we look at this process, we can really break it down into three different steps. You need to have your start, you need to have the part where you're making your RNA primer, and then you need to have the end of the process. And just like in DNA replication, where there is an origin of replication that tells the cell where to start, there is a set of signal sequences built into the DNA that are going to indicate this is the start of my gene. And these are what's known as promoters, and they're just, like I said, short little sequences in the DNA there that the enzyme RNA polymerase is going to be able to read that, and that tells the enzyme, hey, go ahead and start making a copy of this DNA, because that is going to be what we need to read to tell us, or to basically provide those instructions for the cell with what to do with that. So remember, our definition of a gene is that it codes for a functional product. And in this case, with transcription, it's coding for an RNA copy of the DNA. Your RNA polymerase is going to be coming along and binding to this promoter region on the DNA. And the RNA polymerase, when we look at it in E. coli, it actually does have 6 different proteins that are going to bind together to form your holoenzyme. And one of those proteins in our E. coli is known as sigma factor. And this is the part that actually binds to your DNA promoter right there. And so this is the part of the RNA polymerase that can read the DNA to tell it, hey, this is the start of my gene. And one of the other proteins on that RNA polymerase holoenzyme has a helicase activity. So in DNA replication, we saw that DNA helicase was separate from our polymerase. Well, here, our RNA polymerase actually brings along its own helicase. And that helicase is going to start unzipping your DNA, and it's going to give you something that was reminiscent of our replication bubble for our DNA that was out there. The RNA polymerase will unzip the DNA molecule there, and then you're going to go into your elongation phase, and that's where the RNA bases that are complementary to our DNA are going to start lining up on our DNA right there. Again, you're going to form those hydrogen bonds between those complementary bases. So if we looked at this, the next RNA base that would fit in there is the one that base pairs with A. So you're going to have a U, and then you'll have an A, and then a C, and it'll keep going down. So if you think about that, you're effectively lining up all of your bases in a row, and your enzyme is going to come along and burn the energy in those nucleotide triphosphates, again, ATP. And when they hydrolyze those phosphate bonds, that energy is going to get released, and it's going to allow our RNA polymerase enzyme to make that phosphodiester bond and give us our sugar phosphate backbone between those bases there. At the end of this process, the RNA polymerase is going to run into another signal that's built into the DNA, and this one is known as the terminator. And that's going to result in your RNA polymerase enzyme falling off your DNA. Your 2 DNA strands are going to re-anneal, and your DNA is going to be perfectly good, because the only thing that's happened to it is the 2 strands have separated, and then they've come back together. But you have, in the meantime, made a copy of one of those strands of your gene, and now you've got this RNA copy that can float around in the cell, and it has its own function. And if we look at the RNAs that can be made from your DNA, these are the three basic ones that we will find out there. One type of RNA that is made during this process of transcription is known as messenger RNA. And these are typically going to be about 1,000 bases long, and they are not very stable. They will get degraded inside the cells relatively quickly, but their function is this is going to carry the message out to the ribosome that tells the ribosome here is the amino acids that need to get hooked in together to make a protein. The other kinds that we can find is a transfer RNA, is another RNA that can be made from genes on the chromosome. These guys are much shorter. Rather than 1,000 bases, these guys are typically less than 100 bases, so 70 to 100 nucleotides long. They're really stable, unlike your message, and they are really stable because they actually fold back onto themselves. And so you can see they actually do make hydrogen bonds between complementary sequences within that molecule right there. The reason for your tRNA or transfer RNA is on the 3' hydroxyl end of this thing. That's where you're actually going to get your amino acid, and I'll show you a picture of that in a second. But our last major type of RNA that we'll find in cells is known as ribosomal RNA. And these guys are going to be a lot longer, a few thousand bases long. And if you were to look at a ribosome, about 60% of this enzyme is actually RNA molecules there. So the RNA makes up what's known as the scaffold, and then your proteins are sort of arrayed all over the scaffold to give you your holoenzyme there. And the ribosome's function is it's going to recognize the message, and it's going to provide a site where the tRNAs can bring your amino acids together, and the ribosome will actually make that peptide bond between those amino acids. And so we'll take a look at that in the next step. But first we want to take a look at the structure of your tRNA molecules here. And so here we can see our tRNA molecule that's been folded up over on itself, and on this 3' hydroxyl end right there, we have our amino acid. And so when you look at this, this is known as the cloverleaf diagram right here. But these tRNAs actually sort of fold over on top of themselves. And so if you look in three-dimensional space, this thing would almost be folded in half right here. So there's your 3' hydroxyl group where your amino acids would be attached to the tRNA. And that 3' hydroxyl group, just like it's important for DNA or RNA synthesis, this is the functional group where an enzyme that is known as an amino-acyl tRNA synthase is going to add an amino acid onto that 3' hydroxyl so that your tRNA is basically ferrying or carrying that amino acid around in the cell there. The other important part of our tRNA molecule is what's known as the anticodon. That has these three bases that are sticking out here or sticking out over here. And if you look at the process and what goes on with this, those tRNAs actually are going to complement some bases in the message. So think about your messenger RNA. Well, the message that is encoded in that messenger RNA, all of the words are actually 3 bases long, and they're known as codons. And so these codons right here are going to match or complement your anticodons in your tRNAs there. And those tRNAs are going to be able to bind or form those hydrogen bonds right there with the codons in your message, and the tRNAs are carrying those amino acids. So if we think about this, your tRNA can actually read nucleic acids, and it can read amino acid over there. And those tRNAs are going to carry those amino acids to the message right there. So effectively, the codons in the message are going to determine the sequence of amino acids in your eventual protein there. And if we look at this process here, think about codons in message are going to be equal to codons in your tRNA, and that's going to be equal to the amino acids. And if we look at this process here, there is a thing out there known as the genetic codon table, or the genetic code. And this is something that was actually discovered by Francis Crick after he got the Nobel Prize with James Watson. He went to go ahead and look at the process for what is the DNA used for inside the cells. And he was able, in his lab, and a lot of people working in it, were able to figure out the genetic code, or the codon table right here. And this shows you the 3-letter combinations of A, C, G, and U in all of their different possible combinations. So you could have 4 different bases in the first position, 4 in the second, and 4 in the third. And if you take the number of possible combinations, 4 times 4 times 4, you get 64 possible combinations there. And so what we can see here is all the possible combinations of codons and the amino acid that corresponds to that. So this would effectively, this little parentheses thing right here, that would be like your tRNAs that are binding up to the codons in the message. So if we look at this first codon right here, this reads AUG. There's a tRNA out there that has the complementary sequence to that AUG right there. And it's got this little thing. But remember, this is going 5' to 3' right here. And so if we look at this process right here, we're not going to look at our anticodon. We're going to look at the codon right there. And AUG corresponds to this little guy right there in our codon table. And the first amino acid is a methionine. The second codon right here is GGA. So we come over here and we scan through. There's our GGA, and we've got an amino acid called glycine. And so we just work our way through this codon table right here, and you can actually take a message and use that to determine your amino acid sequence right there. The other thing that we need to take into consideration is when people looked at the structure of the ribosome in a lot more detail, they found there is 3 pockets, almost like different rooms for different activities built into the ribosome there. And here we can see a ribosome that is clamped down on a message, and the ribosome is basically going to move along the message here from that 5' end to the 3' end. So over and over we see that cells have evolved, so there is some directionality to what we're looking at here in terms of the roads here. The ribosome only goes in one direction. It does not read the message from 3' to 5' end. It does not work that way. But if we look at these different pockets here and start to take a look at what activities are associated with them, going from this side, and you'll see why, your tRNAs are actually going to enter your ribosome through this side over here, and so the tRNAs are going to come in over here carrying the amino acid. So there is our A. The A site actually holds the amino acyl tRNA that has the tRNA bound to that amino acid. And so your tRNAs are actually going in this direction when your ribosome is going in this direction. The second pocket, when we take a look at the middle here, is the P site, and this is where the ribosome actually holds the short peptide, or what's known as the peptidyl tRNA. So if you have a tRNA that is bound to that codon right there with its RNA, the peptide is going to be held on that tRNA. And then the third part of this is the E site, and that's this little guy over here. This is where the tRNA is going to remain stuck to your message right there, and it's going to exit. So thus we have amino acid entry, we've got peptide, and then we've got exit. And so your tRNA is going to come shooting out of here, and it can float around in the cytoplasm for the cell, where it's going to get recharged by another one of those enzymes that adds that amino acid to the 3' hydroxyl group there. And so we can take a look at this process of translation where we have to put all of these things together. And just like with the other things, we can see that it has to have somewhere where it starts. It has to have an enzyme activity that is going to make your polymer, your long repeating chain, in this case of amino acids, and then you have to have something that tells it where to stop that process. And so we'll take a look in the next video at this process of initiation, elongation, and also termination. All of them are steps in this process of translation where we're going from a nucleic acid to an amino acid. And so again, there's that pun. Translating is going from one language to another. Here we're going from the language of nucleic acid to amino acid.